you. Thank you very much, Hao, for the introduction, and it's a great privilege to be here on this stage. So I would like to talk about geometric deep learning and the applications of these methods to the field of life science and biology. And I guess we all have our scientific heroes, right? I for sure have. So imagine yourself a student coming to meet your hero in science, a very prominent researcher, and unknown to you, his place is taken by the devil, who speaks to you in this way about his view on science. And he says that who traces life and seeks to give descriptions of the things that live, begins with killing to dissect. He gets the pieces to inspect, the lifeless limbs beneath his knife, all parts but link which gave them life. So I'm quoting it in the English translation, but probably you have recognized the famous scene from Goethe's Faust, where Mephistopheles pretends to be Faust himself and speaks to a scholar about actually what I think is a poetic description of the problem that we encounter in many fields of science, but in particular in the field of life science and biology, which is the problem of scales, right? So if we take a living organism, we can look at it at different scales. We can look at the individual molecules, right, that act on nanometer and femtosecond scale, all the way to cells, tissues, organs, and maybe entire ecosystems that are more than 10 orders of magnitude apart in the spatial and temporal scale. And this creates a big problem because we usually have a very good understanding of the lower scale. So the molecular scale, how different atoms interact in order to form a molecule, right? We can describe it very accurately using models in quantum mechanics. But as we go to the higher scales, to higher levels of abstraction, we usually have no clue, right? And this is, if we look at the typical molecule, right, that small drug contains maybe 10 or 20 atoms, we can very precisely describe it. If we go to a bigger molecule like a protein, which might have 10,000 atoms, we already cannot use, for example, quantum mechanical descriptions. So we need to use different mathematical techniques, different level of abstraction, and this is just one protein, a single cell will contain something like 100 trillion atoms, and we have more than trillion cells in our body, right? So just scaling techniques that work at the lower level to the higher level of abstraction is totally impossible. And I think it was very nicely captured by Philip Anderson, Nobel laureate in physics, who was saying that the ability to reduce everything to simple fundamental laws doesn't imply the ability to start from these laws, and instead at each level of complexity, entirely new properties appear. And physicists call these emergent phenomena, and this is what actually makes physics work. The problem is that if we take any computational science, right, from designing cars and airplanes to forecasting weather, predicting climate, or developing new drugs, the very premise of these approaches is that we start with some model of the problem, right, or the system that we are trying to analyze. Typically, it's written mathematically as a system of differential equations, right, that we can then program in software code, put in a computer and run this code on some data and produce a desired output, right? So this is how software has always been developed. Now, the problem with biological systems and complex systems in general is that we have no idea how these laws look like. We cannot write an equation of a cell, right? So what we need to do, and probably this is the reason why AI and machine learning is the only hope for addressing these kind of problems, we need to reverse the order. And this is what I think very aptly Andrej Karpathy called software 2.0. So instead of giving the code explicitly in the form of machine instructions, we give examples of data, inputs and outputs, and then with appropriate computational complexity and appropriate data scale, we can figure out implicitly the code that implements this mapping from the input to the output. And in deep learning architectures, this typically takes the form of the weights of the neural network, right? Its parameters, and some of the big models have billions of these parameters. So what we lose in this way, of course, is some interpretability. We don't have a good understanding of how these models work. That's why they are often called black box. But what we do gain is that it is very hard to describe phenomena at certain levels of abstraction, so we don't describe them in a handcrafted way. We try to model them from the data. And this is one of the reasons why in applications of AI for science, deep learning methods have been so successful across the board. So there have been groundbreaking discoveries from biological applications to weather forecasting to pure mathematics, proving new theorems with the help of AI. Now, I know that I'm in a computer vision conference, so I need to show a vision example. So take a problem like detecting cats and dogs in an image, right? So we don't have an equation that describes how a cat looks like, right? It's very difficult to describe what makes a cat look like a cat. But what we can do, we can show examples to a neural network, right? To this black box that contains a lot of different knobs, a lot of parameters that we can optimize for, and that's what we call the training. And we try to generalize from examples that we've seen to examples that we have never seen before, and this way we can deploy this neural network in applications and make it work. The problem is that the number of examples, if you approach this problem mathematically as some form of function approximation, grows exponentially with the dimensions. So we run into a problem that mathematicians call the curse of dimensionality, and the number of cats and dogs that I will need to show to such an architecture, because they are very high dimensional data points, will be not only bigger than the population of cats and dogs on Earth, but probably more than the number of atoms in the universe. So the way that these problems were dealt in the history of machine learning and deep neural networks, actually the inspiration comes from neuroscience, from the seminal work of Hubel and Wiesel on the organization and the function of the visual cortex of the brain that brought them the Nobel Prize in Medicine in 1981. And these ideas were implemented and mimicked on a computer, first by Kunihiro Fukushima in the early 80s, and then famously by Jan LeCun in the late 80s in the convolutional neural networks. And the main idea that distinguishes convolutional neural networks and actually make them work practically is these two notions that you work locally, so the neurons are connected only to what is called receptive field, and you also share parameters. And this way you can reduce the number of degrees of freedom from quadratic to constant, and you can tame the curse of the dimensionality. And doing this also without maybe explicitly acknowledging it at first endows the architecture with the geometric property that we call translation invariance. So roughly speaking, this is to say that no matter where the cat is located in the image, I want still to be able to say that this is a cat and not something else, right? And as you probably know, the computer vision community initially was skeptical to these ideas. So if we look at how computer vision pipelines in image recognition looked like 20 years ago, it was extracting handcrafted features, feature descriptors, and then applying some simple classifier to them. Everything has changed in 2012. It was a convergence or a confluence of trends of the growing compute power, in particular the GPUs, availability of large data sets, and somehow re-engineered and reinvented architectures, the same convolutional neural networks that allowed to significantly outperform anything that has been handcrafted. And I'm talking about the ImageNet breakthrough, this moment of surprise for the entire community that has changed dramatically the way that computer vision is done and also created new multi-billion industries and multi-billion companies. And the rest is history, right? 2018, the Turing Award was given for deep learning. And now this is a technology that is probably one of the most important technologies invented by humankind. Now, talking about biological applications, let me give you another example of data. So this is a molecule represented as a graph. I guess we all like this molecule. This is caffeine, right? You probably had a lot of it already these days. So one of the problems that we need to routinely solve when we, for example, do drug design and drug discovery is to predict computationally properties of potential drugs, right? We cannot test everything in the lab, so we need to do it computationally. And the problem here, again, if I want to feed this kind of input to a neural network, I have the problem that I don't have a canonical way of ordering the atoms in this molecule, right? So it can come up to any permutation. So we need another geometric property that we call permutation invariance. And this is to say that the molecule doesn't know how I decided to order its atoms on the computer. It's basically some kind of arbitrary representation. Now, the molecule itself is also a geometric object, right? So these atoms are not just abstract nodes of a graph. They live in three-dimensional space, so I can position the molecule differently in the space, so I can subject it to transformations such as rotations and translations. We still want to be able to predict these properties correctly, and this is, again, a geometric property that we call SE3 invariance. SE stands for the special Euclidean group, basically operations like translations and rotations. And I guess you get the point. So the general umbrella term for these kind of methods is called geometric deep learning. So it is a principled, disciplined way of designing neural network architectures that allow us to address appropriately the transformations that the input can undergo. So I can guarantee that the neural network will output certain things when the input is subjected to the action of a certain group. And the mathematical principle that underlies these ideas is the notion of symmetry, which, as Hermann Weyl, one of the most important mathematicians of the 20th century, was poetically referring to as this one idea by which man through the ages has tried to comprehend and create order, beauty, and perfection. So it was fundamental to ancient Greeks, who are credited with the invention or formalization of geometry. So the Euclidean geometry stood more than 2,000 years until it was challenged in the beginning of the 19th century with discovery or invention of other types of non-Euclidean geometry. And the revolution in geometry, the systematization or reinvention of geometry by Felix Klein, the so-called famous Erlangen program, is again based on the concept of symmetry. So the idea that you can define geometry as the properties of objects when they are subjected to certain geometric transformations. For example, Euclidean geometry arises from rigid motion, the Euclidean group, and you can generalize it to many other types of transformations. So the same very ideas in the beginning of the 20th century came to the domain of physics, where physicists realized that the very laws of the universe could be derived mathematically from these first principles. So, for example, when we talk about conservation laws like conservation of energy, it's not just an empirical observation that you measure in an experiment. You can actually derive it mathematically from the symmetry of time. So that was famous result, the Noether theorem, and it has changed dramatically the physics that we know nowadays. And again, the same Philip Anderson that I quoted before said, I think very laconically and very beautifully, that physics is the study of symmetry. Now, if you're familiar with physics, you would know that this is how we see the universe, right? So this is the standard model. It all boils down to symmetry groups. So the Minkowski geometry of the space-time that is derived from the Poincaré group, or the different fundamental forces that act on fundamental particles, they come from the symmetries of the quantum fields. And you can see that if symmetry underpins modern physics, it also underpins deep learning. So in this example of a molecule, we have two types of symmetries, the permutations of the atoms in the molecule and the continuous transformations, the special Euclidean group. And these architectures, so they appeared relatively recently, a few years ago. Probably the most famous instance of this kind of geometric thinking and these geometric architectures was DeepMind's AlphaFold, the architecture that famously predicted three-dimensional structures of proteins. And in particular, they use what they call invariant point attention, which mathematically speaking is an equivariant transformer. Now, why proteins are important? So you may think that if you're not in this field, proteins is some stuff that you take to bulk muscles. But actually, all life that we know about is based on proteins. We are not familiar, at least for the time being, with the life form that is based on something else. And proteins literally are responsible for anything that is important in our body, from defending us against microbes, right? Antibodies are special types of proteins, delivering oxygen to our cells. Even any chemical reaction that happens in our body would not be possible without catalysts that are called enzymes that are, again, special types of proteins. Now, chemically, what proteins are, there are sequences of basic building blocks called amino acids. So these are long chains of biopolymers that, under the influence of electrostatic forces, fold into complicated three-dimensional structures. And it is this structure that endows the protein with its function, the ability to bind to other proteins or to other molecules in our cell. And it was conjectured already in the 70s by another Nobel laureate, Christian Anfinsen, that it is the sequence of amino acids that determines in certain conditions the structure of the protein. And it has become one of the holy grails of structural biology, how to predict the structure of the protein given the sequence of amino acids. There was a competition called CASP, established for this purpose since the 90s, and different scientists, different groups were competing who predicts better the structure, a little bit similar to the ImageNet competition in computer vision. And structural biologists have had their own ImageNet moment when a deep learning approach from a team that has never been active in the space of biology, DeepMind, who has ever heard about them, significantly outperform anybody else and there was first the AlphaFold, then AlphaFold 2 that already reached the quality of experimental data. And again, you can say that the rest is history, even though history is being written in these days. The Nobel Prize in Chemistry was given last year to the DeepMind team for AlphaFold 2. Now, it is interesting to look at these two breakthroughs that happened in the field of AI, ImageNet in 2012 and AlphaFold in 2022, exactly a decade apart, and to try to draw some conclusions, right? So what would be the next breakthrough and how to make it? And there is this somewhat controversial argument in machine learning that data is more important than algorithms. And I think it was first conjectured by Alex Wisner-Gross. He looked at the time that it takes from the introduction of an algorithm that has led to a breakthrough versus the introduction of a data set on which the algorithm was trained. And the trend was always in favor of the data. So in case of ImageNet, for example, CNNs existed for more than 30 years. ImageNet dataset was introduced 2 years before the AlexNet paper was published. It is interesting, actually, that the trend is reversed in the case of AlphaFold. So the methods on which it is heavily based, transformers, right, 2017, equivariant architectures, roughly the same time. The data sets have been built since the 70s, right, especially the structural data, protein database, or the PDB. So this brings another question that is often overlooked, the selection of the problem. And it's a question that probably we should all be asking ourselves, what is necessary to make machine learning successful in a particular biological problem, but a problem in general, right? And I wish I knew the answer. I think I can only hypothesize. I would say that there are at least 4 ingredients. 2 of them are probably non-controversial, and others we can argue about. So the first 2 are availability of large and diverse data sets and clear performance criteria, where it becomes a competition, like ImageNet or CASP. The other 2 are the presence of internal structure in the problem and what we call degenerate solution space. So let's first talk about the number 1 and number 2 that are less controversial. In case of ImageNet, right, it was established in 2010. It contained 1,000 fine-grained classes, so not only classification of cats and dogs, but particular breeds of cats and dogs. More than 1 million training images that were manually labeled by a service of AWS at that time called Mechanical Turk, and the performance was evaluated as average test accuracy. In CASP, same story. So CASP itself was established in the 90s based on PDB that started already in the 70s. So these are every protein structure that was solved by structural biologists was deposited in this data set. And the gain performance criterion was very clear. The root mean square distance between the atoms of the predicted structure and the ground truth one that was available by crystallographic methods. So the third point, the degenerate solution space, it is a little bit difficult to quantify in precise mathematical terms, so I would resort to an analogy. If you think of the space of cat or dog images, right? So it is a very high-dimensional space. We are typically talking about multi-megapixel images, so each of them can be thought as a point in a million-dimensional space. And yet, these images do not fill uniformly this space. Actually, if you were to visualize, and it's very hard to visualize million-dimensional spaces, you will see that they will somehow cluster along a low-dimensional structure. So mathematically, you can think of it roughly as sampling from a manifold, right, or a hyperdimensional surface. And this is often called in vision manifold hypothesis, that high-dimensional data lives in the low-dimensional subspace. And there are many reasons why this could be the case. So there are theories, for example, about fractal nature of images. So you can take a cat and construct it out of pieces and bits of the image itself, right, under some geometric transformations. So it seems that something similar happens in the case of proteins. So when people try to impress you, they would give you this number, right, 10 to the power of 300. So that's the theoretical number of proteins that might exist in nature, right? So I don't know if you even realize how big this number is, right? So it's bigger than anything you can imagine. Bigger than the number of atoms in the universe, right? So we have only 10 to the power 80 atoms in the universe. It's called, in physics, the Eddington number. If you take every organism, every virus, every bacteria, every horse, every cat, and every human, it will be just 10 to the power 30. The national debt of the US, it's 10 to the power 13 US dollars, right? So everything is dwarfed by this number of proteins. And yet we don't see them in nature. And you can argue why this is the reason. Probably one of them is that nature is lazy and it likes to reuse working solutions. So if something has worked, right, certain protein structure is doing its job correctly, then let's just reuse it in slightly modified shape. And this is one of my favorite examples that I think exemplifies it very nicely. This is called the timbarrel fold. So it's a very ancient protein structure that has been on Earth probably more than 3 billion years. It was likely found in the last universal common ancestor, so this primitive organism from which we all have evolved. And you can see that it's some copy and paste with some modifications of a simple structure. It's called beta-alpha unit. We find it in more than 10% of all enzymes across functions and across species. So more or less any metabolic activity that bacteria, tomatoes, or humans do, you will likely find this kind of structure, right? So it is heavily reused. It is copied and pasted with modifications, and that's why we have this kind of reduced complexity that is significantly smaller than the theoretical astronomical number that we see. So the internal structure, here again an apt analogy would be from the domain of language. And if I give you this sentence and I removed one of the words, I guess you will have no problem finding out what the word was, right? So you can probably find some meaningful completions here, and most likely bank, which in English has also the meaning of a side of the river, would be a correct match. And how would I know this? Because of the context, right? So the context talks about river. If I gave you a different phrase that talks about financial institutions, again, bank has a different meaning in this case of a financial institution, and again, the context tells me that this is a correct completion. So this is a very primitive and very naive description of how models like ChatGPT work, right? So now imagine that you have trillions of such examples, and you train a neural network that tries to solve this task, and by doing it, you learn not only the grammar of the language, which conjugation or inflection of the words to use, but also what is the language semantics, the right meaning of the word. And this is possible because of a property that is called distributional hypothesis, saying that words that occur in the same context tend to have similar meanings. So the same thing happens in proteins. In proteins, we have evolutionary history. So proteins exist across different species, across organisms that are related in the branches of the tree of life, in the tree of evolution, and they have conserved structures. So if something is important for a certain biological function, it is kept and is not mutated, because if it mutated and the structure broke, then the organism would not have survived. So AlphaFold also heavily leverages this kind of principle in order to work. So all summarized, do we actually have a theory of protein folding? So can we say that AlphaFold 2 has learned the physical principles that underlie the creation of this beautiful three-dimensional structure? And there are arguments both ways. I tend to believe that it doesn't, and people from DeepMind probably say that the other way around, but I think it's the wrong question. I think we should ask the question whether we have to learn the physical laws of protein folding, and this brings us maybe to a more philosophical question, is whether the science in the age of AI will be done in the same way as it has been done before. And the person who popularized this idea of post-theory science was Chris Anderson, at that time editor-in-chief of the Wired magazine. And here I think it's beautifully captured in this quote that between Newton and Zuckerberg, theory took a backseat and so much data had accumulated, and computers are already so much better than us at finding relationships within it. Soon the old scientific method of hypothesize, predict, and test would be relegated to the dustbin of history. Now, whether you take it at face value or with some skepticism, I think it is true that science has worked in the same way today and in the time of Newton, right? So it was always these steps of hypothesize, predict, and test, and technology has been used at different stages of the scientific process. Newton himself invented a new device, a refractor telescope that he used to test his predictions of planetary motions. With the advent of digital computers, all the predictions are made in simulation, right, on a computer, but the hypothesize step has always been the output of a human brain, right? The eureka moment, the spark of genius, the creativity, and we are living for the first time in history of science and history of humankind that the machine is eating our bread, that is becoming increasingly part of the creative process of scientific discovery. And I don't think that human scientists will disappear anytime soon, but I think we need to revisit the way that science is being done in the age of AI. And probably the first thing that we need to ask is whether the data sources are still adequate when the consumer of the data will be artificial intelligence. And if we look at AlphaFold, right, everybody now is trying to think how to make the next AlphaFold, right? And I'm using it as a metaphor. In general, algorithms that would solve biological problems. But in my view, the overlooked problem is how to make the next PDB. What would be the next data set, the next data source? And AlphaFold, by machine learning standards, used a tiny amount of data compared to, for example, models like OpenAI ChatGPT. And yet it took more than 50 years and an estimate of more than $50 billion to create this data set. So if we want to make the next breakthrough in this field, we are missing probably at least 6 orders of magnitude in terms of time and cost. And it's a good question, what would be this next data source? And this is something that we call black box data. So it's a little bit controversial term because black box has a negative connotation, but if we take the problem of protein structure prediction, you can argue that the early methods were physics-based, right? So they came from first principles. So David Baker, who was one of the co-recipients of the Nobel Prize last year, is famous for Rosetta. So it's physics-based simulation of how proteins fold and how proteins can be designed. So it's white box software 1.0. So the breakthrough that came from DeepMind and similar approaches was to replace the white box with the black box. We don't really understand what it does, but it does it better. And some biotech companies were able to use these approaches still with traditional data sources, just scale them. And most of the sources in biology are either based on sequencing or based on imaging. I think that the next logical step would be to move away from data that is a white box data that is understood by humans to data that makes no sense to a human without appropriate machine learning algorithm. We call this black box data. It is not completely new idea. You can actually find instances of these type of data that makes no sense to humans in the purest mathematical form, for example, in the field of compressed sensing, where the data literally looks like noise. But what it allows is to use significantly cheaper, high-throughput technologies to acquire the data and to co-develop machine learning methods that will be using this data. And one example that I would like to give, so this is from a company where I'm chief scientist. So VantAI is a company in the business of designing molecular glues. So molecular glues are small molecules that enable interaction between proteins that otherwise do not interact. And basically, interaction between proteins is responsible for everything that we have in biology, right? So that's how cells operate. This is how disease is caused, right? Because certain things interact where they shouldn't be interacting. So the source of data that VantAI is using is structural proteomics. So it's a mass spectrometry-based technology that allows not to get the full three-dimensional protein structure, but to get a sparse, coarse interaction between protein interfaces. And this data itself, this technology has existed before, but it was not considered sufficiently good for these kind of applications. So this is the first model that we developed called Neo-1. So it's a multi-model foundational model that allows to take different data sources, whether it's sequences, whether it's partial structures, whether it is kind of black box data, and at least to my knowledge, for the first time to do simultaneous co-folding of protein complexes and de novo generation of the small molecules. And this is a kind of chicken and egg problem because the proteins do not form a complex without a small molecule, and you don't know how the small molecule looks like. And so this is an example of this sort of black box data. I'm pretty sure there will be more other data sources. And what it does from the perspective that I presented before, it reduces significantly the search space. So it makes a problem that is otherwise intractable into a problem that is more tractable. And we see many examples. So this is a complicated structure again, so some form of molecular glue that, for example, programs like AlphaFold 3 are unable to predict correctly. And with these kind of structural constraints, we are able to predict significantly better, and it seems to be working across the board very efficiently and very accurately. So I started with a quote from Goethe, and I would like to finish with a quote from the same great poet. You know him as a poet, but he was also a scientist, or as it was called at that time, a natural philosopher. And he actually studied chemistry in Strasbourg with Spielmann, who was one of the last generation of alchemists. And alchemists, when they didn't know how things work, especially how inanimate things make living stuff, they resorted to magic. So some kind of semi-magical forces that make things move and live and reproduce and so on. And they called it encheresis naturae, the hand of nature, that pressed life into organisms. And Goethe apparently was very unhappy with this explanation because it didn't explain anything. So he put famously this term into the mocking mouth of Mephistopheles. And in the English translation, it's encheresis naturae is the chemical name by which dunces themselves unwittingly shame. So it's more than 250 years since, and we don't need to resort to magic anymore. We actually have much better understanding of how molecules interact and how cells work. But I think it's a warning, because overly relying on AI to somehow magically solve our problems in life sciences, in biology, in science, more generally, is a return to alchemy, or this time maybe encheresis machine, the hand of the machine. So I think that if there will be breakthroughs in this field, they will come not only from some magic power of AI, but from combination of new wet lab technologies, in particular new data sources that are co-developed and co-designed with artificial methods that are able to take advantage of these new data sources. So I will stop here. Thank you very much for your attention.
Is there Q&A? Okay. All right, so are there any questions? Well, we're going to have a little Q&A sessions with Michael. So if anyone has a question to Michael, please raise your hand. Yep. I think we have a question over there.
Thank you, Professor Bronstein, for the great presentation. So I have the question regarding the data, regarding the black box. What's your vision on the synthetic data set? Because, you know, some of the, less complicated interactions could be designed using physics, and which could be used for as a training set. Thank you.
Yeah, very, very good question. So synthetic data, right, or simulations. So this is actually, well, bread and butter, especially in biological science or chemical sciences. So we do it all the time, right? So in case of proteins, this is molecular dynamics. So you can argue that approaches such as molecular dynamics are solving the wrong problem in the right way, right? So what is molecular dynamics? You cannot model a big molecule like a protein on a quantum mechanical level. So you resort to classical model with a lot of tricks that are some kind of black magic, right? And because of numerical considerations, you need to, you want to simulate phenomena that happen at sometimes millisecond scale, right? Like the way that proteins fold or the way that proteins bind. But you need to take steps at the level of femtoseconds. So that's why it can run for a week on a supercomputer. Now, and in many cases, you don't even trust the outcome of the simulation. So if you compare it to generative models, so generative models solve the right problem because they don't really have any constraints on, they don't explicitly model the mechanics of for example, protein folding or protein interaction, but they solve it in a way that is sometimes correct. So an analogy from image generation would be humans with 6 fingers, right? So what we need to be able to do is to be able to quickly discard incorrect solutions. And then I can just continue producing, continue sampling from my distribution, right? So which is what generative models do, until I am satisfied with the solution. So in many cases, this can be done computationally, and that's why I think that formal fields like mathematics will actually be the first where generative AI might replace humans, right? Like theorem proving, which is a little bit paradoxical because this is considered the hallmark of human genius, right? So mathematics, the purest of sciences. But the issue is mathematics is that it's all software, and software is soft, right? So I can, if I have a model that produces a candidate proof of a theorem, I can formally verify whether it's correct. And if it's not correct, I will produce another proof until it is correct. So even if it's 1 in a billion probability to get it correct, I can just crunch numbers until I somehow sample and cover all the space of all mathematical proofs, right? In biology or in life sciences, or I think in general in science, Unfortunately, it's hard to do it computationally, so you need to resort to some experiment. But then if you can design an experiment in a way that is just for this purpose of verifying a solution of some simulation, you can make it a very high throughput, very highly parallelizable, very cheap technology. So instead of testing a single hypothesis at one time, you will be able to test multiple in parallel, right? So I think simulation, maybe approached from this perspective, is a very promising direction for producing data.
Good afternoon, sir. Thank you for that illuminating presentation. I'm a novice. I'm going to ask simple questions like, is there a cure for cancer around the corner? Can asthma be cured? And then your slide was very interesting, geometry. And then we talked biology. And this is the age of combinatorial intelligences. How can we probably fast-track pathology? How can we fast-track prognosis and diagnosis and those kinds?
Yeah. So I'm not a biologist, I'm not a clinician, so it's hard for me to answer the question about cancer. So cancer is not a single disease, it's a class of diseases in which certain processes go wrong. So I would say you can find some common traits and be able to cure them. The problem with cures, basically, it takes roughly 10 years from the moment you have a candidate drug, and I probably will hear from Roche more about these processes, until you actually can use this drug in the clinic. So the drugs that are being developed now, we'll see them in the market in the 2030s, unless the processes like FDA approval and clinical trials become significantly faster, which is also a possibility with the help of AI and machine learning tools. Now, whether we can cure cancer, probably no, but not, at least not immediately, not now. But you can already see there has been some breakthroughs on new therapeutic modalities, so some diseases that a decade ago would be a certain death sentence for the patient, nowadays can be completely cured, right? And in particular, what has been interesting is biological drugs. So the drug, when the molecule itself is a protein or a peptide, so not the regular small molecule, but something biological. Now, about geometry, well, it's a little bit difficult without getting into the details to explain why everything is geometry, right? So it can be very down to earth and something like three-dimensional transformations of molecules, right, which is something that you can visualize. It can be also something very high-dimensional and very abstract, like, for example, symmetries that we have in the space of parameters of a neural network, right? So this is another interesting thing, because the way that we train neural network has been always by some optimization techniques called backpropagation. We can potentially generate parameters in the parameter space directly, accounting in the correct way for the symmetries that they have. Yeah, without any gradient descent. Yeah, exactly. So without backpropagation. So this is something that has been known for a while, but probably for the first time in the last couple of years, it has been exploited properly in a formal way. I think there is a microphone coming.
Thank you for the great talk. So I just want to go back to one of your points where you are basically saying that the deductive closure is going to be achievable by these AI models. So as we all know, there are two sides of a scientific process: deductive closure and inductive leaps. And many people think that inductive leaps are much more attainable by AI models. So, given the Gödel's second incompleteness theorem, I'm wondering, how did you come to a conclusion that actually math is gonna crack first?
I didn't, so probably I was not clear. No, I don't want to make these bombastic claims. So I think there are some opinions of this kind. So I don't have a firm position on whether it would be possible. I think it's a very ungrateful job in this field to make any predictions. You can be contradicted in a matter of a few weeks by some new technological breakthrough. No, so I think I have maybe more cautious view on this. So I think we will not get rid of humans in scientific discovery process anytime soon. It will probably be a new level of abstraction. And probably a good metaphor here is software engineering. So 70 years ago, we programmed computers writing binary code, right? And then came the assembly languages, and then functional programming, and then object-oriented programming, and as one of the previous speakers said, now the most fashionable programming language is becoming English. So it's not that the humans will disappear, right, or software engineers will disappear. They will be working at a new level of abstraction, most likely more productively. Probably in the next years, we'll see possibility to develop software products without any coding, so it will democratize this process to people who otherwise would not be able to do it, which of course also implies that the entire industry will undergo tectonic shifts.
So you mentioned these foundation models, and as you know, as a data practitioner, we are working in different various problem segments, like autonomous driving or healthcare sector. When we are working with the AI agents, people or AI practitioners, they are saying that we are very close to like 3 years to super intelligence, just one foundation model. But I know that they are just trying to attract investors. As a scientist, what do you think? We are very close to the super intelligence, one foundation model can deal with the different problems, or it's just imagination for now?
I don't know. So I think the definition of super-intelligence also varies, and it's hard to say even what constitutes an intelligence, right? So I'm not sure if it's even the right category, and it's a necessary category. So it's a kind of, I view it as a line of the horizon. So the closer we approach this goal, somehow it moves away from us, right? So we are, for example, already several years beyond the Turing test, and yet we don't have general or super-intelligence, right? So it's probably not very relevant question. So I think for a particular field of application, if you can define some performance criteria, and there are some, right, and you probably need to constantly update them because machines manage to crack them continuously. Yeah, so I think it's a question of definition more than some particular event that will happen, in my view. Yeah, I can hear. Yeah, well, probably Hao would be able to answer this question better. I haven't been working on computer vision for quite a while. I think what we have seen in the last years is very close to science fiction, even for those who have been working in this field. So the progress has been dramatic and very fast. So I would say that, well, even if machines cannot see yet, right, and depending how you define it, I would say that machines can see. So many tasks are already beyond human capabilities, or significantly faster, significantly better. Probably take more or less any task and let it a couple of years and it will be solved, in my view, with enough computational and data scale.
Maybe just some final comments. I think you gave a really amazing presentation on something that I think will really deeply impact even how research, scientific discoveries, and artificial intelligence is going forward. I think we're really living in like a time where things are completely transformative. With my university, we're working on a new kind of undergraduate degree in artificial intelligence. We have to completely rethink about what the future is going to happen.
Totally. Well, and you're doing it now in MBZU AI, so I think that's a great effort actually when you can do things from scratch. How would you imagine the new education in the future of AI?
Right. So maybe just before you go off stage, can you give us some, takeaway messages from your presentation, and maybe some of the key highlights that you think is the most important thing for, for the audience to remember?
well, I don't know. I'm a big believer in the fact that principles are more important than their specific instances. And yeah, I think it was Helvetius who said that being able to understand principles compensates for the lack of knowledge of facts. So I think if I were to summarize what we are trying to do with geometric ML, and I think more broadly in this community, I would go with this quote from the great Swiss philosopher